So thanks very much for inviting me to speak today. It's really an honor to talk a little bit about our work manipulating tropomycin 1 and iPSCs to enhance in vitro blood cell production. I have no conflicts of interest to disclose in relation to this work, and as we've just heard over the past half hour, there's a pretty significant clinical and translational need to enhance and improve our ability to make blood cells. I'm a clinical neonatology fellow at the Children's Hospital of Philadelphia, and I think from my perspective, I've been pretty focused on the problems with our donor-derived platelet supplies. And to be clear, at least pre-pandemic, the donated levels of platelets had been as high as they ever were through the Red Cross. And between advanced screening, leukoreduction, reduction, and radiation, the platelets that we actually transfuse into patients are as safe as they've ever been. But increased clinical usage has given rise to significant concerns that the supply of donated platelets will be inadequate to meet clinical demand sometime within the next decade. Furthermore, donated platelet transfusions incur pretty significant complications, such as increased infection rates as a result of needing to store platelets at room temperature prior to transfusion, increased rates of alloimmunization, particularly in patients who require repeat transfusions. And from a neonatology standpoint, probably the most concerning aspect to me is an increased morbidity and mortality in preterm neonates who do require platelet transfusions that seems specific to the platelets themselves. So for that reason, there have been a number of potential solutions put forward, and in vitro-derived platelets and blood cells are certainly one of them. So this technology relies on human stem cells being cultured and differentiated into hematopoietic progenitor cells, megakaryocytes, and ultimately transfusible platelets. If put into clinical practice more widespread, you could potentially deal with our limited supply of donated platelets, given the unlimited regenerative potential of human stem cells, and potentially mitigate some of the complications that I mentioned. But one huge barrier to clinical implementation is the massive cost ineffectiveness of these processes. This costs more than $18,000 to derive a unit of platelets in vitro, as opposed to $600 for a donated unit. So for that reason, our group and others are trying to improve this process. And what I can tell you is over the past several years, we've learned that if you spin megakaryocytes in a bioreactor or otherwise induce shear stress, you can increase platelet shedding from them. And over the years, there have been a number of chemical and drug screens aimed at enhancing megakaryocyte maturation and differentiation from human progenitors. We also know a little bit about earlier steps in this process, specifically the fact that biomechanical changes, including through the YAP protein and downstream regulation, these processes can increase hematopoietic progenitor production from stem cells. Although it seems like historically there's been somewhat less emphasis on these earlier stages. And so that's where we thought we could make an impact on the field. And not for nothing, if we could increase hematopoietic progenitor production, that would not only support an interest in increasing in vitro-derived platelets, but you could also potentially support other translational research into deriving red blood cells in vitro as clinical testing reagents or for purposes of transfusion, as well as off-the-shelf CAR T-cell therapy, which we have just heard about. So we started by asking what human genetics told us about factors that regulate these early aspects. And so I'll tell you how we've used genome-wide association studies and cellular studies to show a role for tropomycin 1 in hematopoiesis. So over the past decade, there have been no fewer than 4 very large-scale genome-wide association studies that have sequenced hundreds of thousands of healthy volunteer donors, identified thousands of genomic loci associated with blood trait variation. Now, these thousands of loci affect things in small ways, like they affect the size and shape of your platelets, they affect the size and count of your red blood cells. And there's some presumption that these loci reflect developmental perturbations leading up to the derivation of those terminal blood cells. Certainly at the outset, there was a tremendous amount of enthusiasm that the insights from GWAS were going to lead to a lot of novel therapeutic approaches. And there's some certainly shining examples of that, but there's some widespread concern that this hasn't come to pass on a large scale. I think there's a couple key reasons for this, one of which is that it's difficult to elucidate active genes and mechanisms related to GWAS loci. But at a more basic level, I'm not even sure that we understand which SNPs matter. And I'll tell you more what I mean by that. So when GWAS report sentinel SNPs, shown in black, these are typically associated with a trait of interest with a very low p-value. But we don't inherit our DNA one base pair at a time. You inherit it in chunks called linkage disequilibrium blocks, which are shown in blue. Now, most of these LD blocks are in the non-coding genome, which means that their biological activity is through the transcriptional regulation of important genes. Some GWAS-reported SNPs would certainly have biological activity if they overlap key transcription factor binding sites, if they overlap key histone marks or other chromatin features. There might be a very clear rationale for the reported GWAS SNPs to be causal for the trait of interest and have biological activity. It's actually much more likely, though, that the GWAS SNPs that are reported will miss the mark, and instead it will be SNPs in high linkage disequilibrium with the reported SNPs that have the true causal and biological activities. Now, importantly, GWAS themselves can't stratify between these two possibilities. Our project was to identify truly causal SNPs and the related genes that regulate blood trait variation in humans. We know something about active non-coding regions since we have quantitative trait locus maps for the human genome. And so-called expression quantitative trait loci, or EQTLs, associate with transcriptional regulation of important genes in various human tissues. This is the sort of thing that's done to the GCHEX consortium. We thought if we could identify key transcription factors and epigenetic marks overlying something like platelet trait GWAS loci, maybe we could do a better job of predicting trait-specific loci and related genes. To do this, we used a penalized regression algorithm called LASSO, which utilizes tenfold cross-validation to identify those chromatin features that were significantly and independently enriched or depleted at platelet trait GWAS loci. So from a set of more than 800 chromatin features, we derived a platelet site signature comprised of 38 chromatin features. Now, each feature was assigned a lasso coefficient, which is depicted on the y-axis of this bar plot. This sort of relates the relative importance of the chromatin features to one another, although I will submit to you that these 38 features were picked from a set of more than 800, and so they're all important in their own right. There was some confidence that we got from the features themselves, in that most of these features derive from relevant hematopoietic cell types, including primary megakaryocytes, primary erythroid cells, and K562 cells, which is a megakaryocyte and erythroid cell line. There were two key takeaways that I want to impress upon you. One of which is that if we were looking for biologically active sites, it seemed like we should be looking for GATA binding sites in the genome. This is because GATA factors are some of the most important features in our model and are known to critically regulate hematopoiesis over decades of study. The second thing we were able to do based on this model was to turn this platelet site signature into a scoring algorithm. So we went base pair by base pair genome-wide and looked for those chromatin features from our signature that overlapped each site. We then added up the relevant coefficients for those features to assign a score to each base pair. It then became very easy to cherry pick the top scoring sites as those most likely to be functional. And it turned out that top scoring SNPs within blood trait variation loci that overlapped GATA factors were things that we could get excited about, and specifically seemed to target EQTLs that regulate important genes. Indeed, we identified specific base pairs in the genome that were known to alter platelet traits in humans. But the key takeaways for me were that maybe we can predict important target genes such that in later cellular studies we can interrogate trait or cell type specific biology. And indeed, the genes that were implicated by our loci did just that. They were enriched for hematopoietic and platelet related genes and pathways. So what I've talked to you about so far is the derivation of a platelet locus prediction model. And indeed, this actually outperformed all prior computational prediction algorithms in terms of accuracy and the specificity related to hematopoietic biology. We made an additional hypothesis, though, and that was based on a model that we composed using the same processes that predicted red cell traits. This model also predicted red cell biology more specifically than other prior computational models. And the additional hypothesis we made, having these two models, was that we wondered whether SNPs that scored well in both platelet and red cell trait models might impact a common progenitor cell for those loci through a shared mechanism, things like MEP or potentially hematopoietic progenitor cell biology. Using all these additional stratifications, we whittled the genome down to about a dozen sites and became particularly excited about one SNP called RS11071720, which was a top scoring SNP in both of these models within a platelet trait GWAS locus and overlaps a canonical GATA binding site. This SNP is also known to be an EQTL for the tropomycin 1 gene. And it's known that people with genetic variation at this site have slightly decreased tropomycin 1 expression in cells and slightly increased platelet count. Based on our analysis, they probably have perturbations in red cell traits too that just narrowly miss genome-wide significance. But the key hypothesis and takeaway from this is that we would hypothesize tropomyosin 1 to be the causal gene related to this locus. Now, tropomyosin 1 regulates actin cytoskeletal biology in all of our cells. So we all have 4 tropomyosin genes, which wrap around actin filaments to regulate actin biology, which is important for things like cell shape, strength, motility, adhesion, even molecular signaling. Tropomyosin 4 is known to be important for thrombopoiesis in both mouse and human models. But interesting, there have been no prior studies of tropomyosin 1 functions in human hematopoiesis. We decided to use a primitive hematopoiesis assay to study the role of tropomyosin 1. So this assay relies on induced pluripotent stem cells, which can be induced to undergo sequential differentiation through primitive streak and mesoderm stages to arrive at an endothelial phenotype. And specialized endothelial cells then give rise to hematopoietic progenitor cells, which can be subsequently differentiated into a number of blood cell types. The iPSCs that we used for our study were reprogrammed from human adult peripheral blood mononuclear cells, and specifically the CHOP10 and CHOP14 lines. So we started by asking when tropomyosin 1 was expressed during differentiation. And remarkably, tropomyosin 1 by Western blot seemed to be upregulated around days 4 to 5 of maturation. This sort of correlates with cells adopting an endothelial phenotype. Tropomyosin 1 stays highly expressed in the stromal endothelial later, but was remarkably absent in hematopoietic progenitor cells, as well as later stage differentiated blood cells. This led us to speculate that maybe tropomyosin regulates some aspect of maturation around days 4 to 5. So we used CRISPR-Cas9 genome editing to specifically delete tropomyosin 1 in those 2 iPSC lines that I showed you. In total, we derived 3 knockout clones from those 2 lines, and what I'll show you is largely data related to the CHOP14 knockouts, but we confirmed all important results in the CHOP10 line as well. Consistent with our hypothesis about an endothelial phenotype, early aspects of differentiation were unaffected in tropomyosin 1 knockout cells. So we looked at cell surface markers, we looked at gene expression at the primitive streak and mesoderm stages, and we looked at differentiation kinetics. And we didn't see any changes before we queried endothelial cells. And it was on days 4, 5, and 6 of maturation that we saw the first changes. And specifically, we saw an enrichment of KDR CD31 double positive endothelial cells in tropomyosin 1 knockout cultures. Later on, we were able to recover an increased proportion of CD43 positive blood cells. And when we quantified the total HPC recovery from knockout cultures, we were able to achieve 2 to 3-fold more HPCs from knockout cultures as compared to wild type. And these knockout HPCs seem to be functional in that they differentiated efficiently into megakaryocytes, erythroid, and myeloid lineages. In lineage expansion assays, knockout HPCs derived just as many megakaryocytes, actually had an expanded population of erythroid cells, and normal to increased myeloid production from each tropomyosin 1 knockout HPC compared with wild type. We also did some functional assays in the knockout megakaryocytes, finding normal to increased functional responses to platelet agonists, convulsin, and thrombin in the knockout megs compared to wild type, and also confirmed normal morphology from the primitive megakaryocytes, as well as normal gene expression by microarrays. And so while I acknowledge that 2 to 3-fold increased HPCs is an earth-shattering, tropomyosin 1 knockouts seem to be one way to genetically enhance in vitro hematopoiesis. And for this reason, we thought it was important to define molecular mechanisms that are supported and perturbed to enact these changes. Because then maybe we could even enhance in vitro hematopoiesis without a need for modification. I mentioned these processes before, but given the phenotypes that we saw, we took a closer look at the so-called endothelial to hematopoietic transition. So in days 4 to 6 of primitive hematopoietic differentiation, in culture, the cells are adopting an endothelial phenotype. And some of the cells become specialized or hemogenic endothelium, shown in blue. These are the cells that are going to detach from the extracellular matrix to become the hematopoietic progenitor cells, shown in purple. These are the things that can undergo proliferation and later hematopoietic differentiation. So we thought that tropomyosin 1 might have something to do with this transition based on the effects that we saw in our primitive hematopoiesis assay. So we decided to look at specialized endothelial production by what's called a limiting dilution assay. So we used flow cytometry sorting of KDR-CD31 double positive endothelial cells, and we fact sorted in limiting dilution in a tissue culture plate. So we put anywhere from 3 to 1,000 cells per well, and we did that in replicates across a tissue culture plate. We then took these sorted cells and cultured them in 7 days in hematopoietic cytokines. And after a week, we did flow cytometry and asked which wells had at least 10 CD43 positive blood cells within them. We then looked at this sort of response pattern shown in red, and we could go across at each limiting dilution for cells and sort of plot a line of best fit based on the response and the number of wells that contain blood cells. Using limiting dilution analysis software, we could then convert this line of best fit to a frequency of specialized hemogenic endothelium in each well. So if our hypothesis was that tropomycin-1 knockout cells would have an expanded population of specialized endothelium, we might expect to get something that looks like this, where the slope of the responses in knockout cultures is sort of twice as steep as wild type. And indeed, that is exactly what we saw. So in knockout cultures, the frequency of specialized endothelium was 2-fold higher than in wild type cultures. We also thought it was important to look at the HPCs themselves. And we asked the question in these assays of, in wells in which 300 or 1,000 cells had been plated, what was the sort of ratio of total HPCs in those wells in knockout cultures versus wild type? We saw sort of a preserved ratio, about 2 to 3 in the knockouts compared to the wild type. And this suggests to a certain degree that knockout HPCs aren't hyperproliferative. So this really restricted the role of tropomycin-1 in these cultures to seemingly the specialized endothelial stage, or potentially the early production of nascent HPCs. So I think one interpretation of the limiting dilution assays is that we're deriving more specialized endothelial cells, and that is entirely possible. But I think it's also a potential interpretation of our results that knockout cultures are supporting a better production of nascent HPCs. So there's a process called anoikis, which is an apoptosis-like cell death that can occur upon separation from the extracellular matrix. And maybe something like tropomycin-1 knockout is inhibiting that process called anoikis. In a complementary set of experiments, we also looked for molecular perturbations related to tropomycin-1 changes. So these are the same flow cytometry plots that I showed you before. And I just am showing them to highlight the 3 populations that we analyzed. So we first analyzed induced pluripotent stem cells. We also analyzed FACS-sorted KDR-CD31 positive endothelial cells on day 4, just prior to the large-scale changes in phenotype that we see. And we also looked at hematopoietic progenitor cells and specifically took non-inherent cells. And we subjected these 3 populations to bulk RNA sequencing. On aggregate, when we looked at these results on the heat map shown here, the cell populations are assorting themselves like you would expect based on the dendrogram, with each cell population sort of segregating on its own, and wild type and knockout cells segregating amongst themselves. At the iPSC stage, we saw 214 genes that were perturbed at an upregulation or downregulation of at least 1.5-fold. But these genes didn't tag any pathways with an adjusted p-value of at least 0.05. This is important to say that our starting material here was essentially equivalent and seemed to be from a phenotypic standpoint as well. We then looked at the endothelial stage and identified more than 1,000 genes that were perturbed in the knockout cultures compared to wild type. And these genes related to extracellular matrix organization and integrin expression. And there's also perturbations in downstream signaling pathways related to extracellular matrix and integrins. We also saw some changes in hemostasis-related genes, which was a blood-relevant pathway. Finally, we looked at the hematopoietic stem cells and identified more than 3,000 genes that were perturbed, and again, tagging the same sorts of pathways related to extracellular matrix organization and integrin signaling, and as well as platelet activation and some hemostasis genes. So on aggregate, what these results are showing is that at the endothelial stage, we're perturbing actin dynamics and also extracellular matrix attachment. This is interesting because cell adhesion-related pathways and cell migration pathways have been previously implicated in specialized endothelial biology in both mouse and human cell types. And I think we've identified some interesting targets from a downstream signaling pathway. I also find it interesting that despite the fact that tropomyosin 1 is totally absent in hematopoietic progenitor cells, we're seeing some downstream changes which could relate to the human GWAS findings. But the key question, I think, based on our results, is how these pathways and specific genes impact the endothelial to hematopoietic transition. And so I just wanted to share one potential hypothesis with you, and that is specifically that RAP1 activation may augment EHT in these assays. So what's known is if you perturb actin and integrin functions, you can impact the activity and downstream signaling related to the RAP1 protein. RAP1 is a GTPase, and activating genes called guanine exchange factors can attach GTP to RAP1 and activate downstream signaling pathways. In several biological contexts, this can support epithelial to mesenchymal transition, which is akin to the endothelial to hematopoietic transition, specifically relating to the anoikis, that process that I mentioned earlier, and specifically related to what's called a cadherin switch. And cells that have more N-cadherin expression may be more prepared to undergo a transition to a non-adherent phenotype. In contrast to the activators, there are GTPase activating proteins that hydrolyze GTP and sort of shut off RAP1 activity. When we looked at the endothelial cell stage, tropomyosin 1 knockout cells didn't have any changes in RAP1 expression themselves. However, a number of activating genes were enriched in tropomyosin 1 knockout endothelial cells, suggesting that they would support more RAP1 activation. In contrast, there were normal to decreased expression of inhibitory proteins, which on aggregate, again, would support more RAP1 activity. And indeed, when we looked at sort of the downstream activators of RAP1 activity, we identified more N-cadherin expression in the knockout endothelial cells. So indeed, they may be more prepared to become non-adherent hematopoietic progenitor cells. So I think that's where most of the phenotypes and interest lies here. And specifically at the endothelial and HPC stage. I think we do have some work to do to make sure that there aren't earlier processes that are affected, but I think in large part, if we're able to define tropomyosin 1 related molecular mechanisms, maybe we can harness this biology to support an increased HPC yield in vitro. And we're also, of course, working to see whether we see similar phenotypes in the definitive cultures. So thanks again for the invitation to speak. I wanted to specifically thank my mentors, Ben Voigt, Deb French, and Stella Chou, for their incredible support throughout this project. And a specific thanks to Gordon Keller and Michael Atkins. Actually, last year at the PCTC meeting, we had a lot of discussions and supporting the limiting dilution assays. So thanks again for your attention, and I'm happy to take questions.